Alright guys, here we are going to talk about our second major class of biological molecules, lipids. Last time it was all about carbs, and today it's all about lipids. So if you remember, just a little bit of review here. When we were talking about carbohydrates, we said that the monomer version of a carbohydrate was called a monosaccharide. And glucose was our major monosaccharide. And when you link together a bunch of glucoses via those glycosidic bonds, we would get either a disaccharide if it was two glucoses, or a polysaccharide if it was multiple glucoses. Now with lipids here, lipids don't really follow that pairing so much. They don't follow that rule so much. You can't join together like three lipids and get a polylipid or something like that. Lipids are actually the only class that don't really form polymers. They don't follow that polymer-monomer thing. They are created in the same way, though. They are joined in the same way via those dehydration reactions. And the special type of covalent bond we create here is called an ester linkage rather than a glycosidic linkage. When we look at the structure of lipids, most lipids, not all, will be made primarily of carbon and hydrogen. So just carbon and hydrogen. And to give you a preview here, I'm going to actually flip to the next slide really quick to show you a pretty standard lipid. Right here on the left, this is a fatty acid right here. Maybe some of you guys have heard of fatty acids, like omega-3 fatty acids. That's what they look like. Lots of carbon and hydrogen. Lots of very, very non-polar areas here. Non-polar areas. Because they are non-polar, they don't interact well with water, so lipids are very hydrophobic in general. Okay. And the three major types of lipids we're going to look at here are fats that we just saw, phospholipids, and steroids. The major purpose of lipids, there are two purposes actually. They act as fuel storage, so kind of like some of our carbohydrates, they're for fuel storage, and they also act as cell membranes as well. So let's take a look at our first type of lipid, fats. So fats are also known as triglycerides. And the reason why they're called triglycerides is because they have three of these things called fatty acid tails. Alright, and one of this thing called a glycerol head. So three, glyceride, or for glycerol head. When we look at the structure of the glycerol head, it's three carbons, so 1, 2, and 3, okay? 3 carbons attached to hydroxyl groups, OH groups, okay? And then we attach a fatty acid tail, one fatty acid tail to each hydroxyl group on the glycerol head, okay? And the fatty acid tail, if we look at it, it is, again, mostly nonpolar. We take the OH from the end of the fatty acid tail, the H from the hydroxyl group on the glycerol, we take out water, and in that enzymatic reaction we join the fatty acid tail to the glycerol head. Do that 3 times and you got yourself a big old fat. Okay. Now, with fats and fatty acids, you can describe both of them this way. With both fats and fatty acids, we can have either a long, straight carbon chain, so long, straight chain, like we do right here, okay, or we can have a kinked chain, a chain that's bent. And when we have a kinked chain like this, it's because of the presence of the double bond between the two carbons. Sorry about that, got interrupted here. So we were talking about that carbon-carbon double bond. Okay. When you have a double bond like that, it'll actually cause the bond will actually cause the chain to kink. That's the technical term. It kinks. I know it sounds kind of dirty, but it does bend. Okay, so in an unsaturated fatty acid tail, you have a bend. In a fully saturated tail, it's called saturated because it has as many hydrogens as possible. There's no double bonds that you could add more hydrogens to. Those fatty acid tails are very long, very straight, stick straight. Okay, so with fats and their fatty acid tails, they can be very, very long. Okay, so we can have fatty acid tails of 25, 30 carbons long, or they can be really short, 7 to 8 carbons long. The glycerol head will only ever have 3 carbons though. And for our purposes in Biology 211, it would be a pretty good idea to, if not memorize the structure of glycerol, at least be really, really comfortable in recognizing it, because it's going to pop up a little bit later when we talk about cell respiration. Okay, let's get a little bit deeper into those saturated fats here. Saturated fats include things like butter, right? like coconut oil. A good chunk of the fats in coconut oil are saturated. And here we have our, let me see if I can get my pen to work. Nope, okay. I'm assuming that you guys can see my little pointer though. Okay. So we have our glycerol head right here, and then here's one fatty acid tail. This is written in the organic chemistry carbon shorthand way. So we don't see all of the carbons and hydrogens written out right here. So this zigzag tail right here is just a bunch of carbons and hydrogens. So we have glycerol head and then one fatty acid tail, 2 fatty acid tails, 3 fatty acid tails. And when we look at the physical space that those fatty acid tails take up down here, they are very, very stick straight. Because they are stick straight, they compact easily, they stack easily on each other, and so saturated fats compress, they are dense, and they tend to be solid. They tend to pack very closely and they tend to be solid at room temperature. Although with this weather, if you leave saturated fats out, it's going to melt. Unsaturated fats, on the other hand, as we just said, they have one of those carbon-carbon double bonds in there, okay? And that causes the chain to kink. And because the chain kinks, this whole molecule can no longer fit and stack very easily on itself. Now, if those of you guys who know some of your organic chemistry, that's actually a cis bond as opposed to a trans bond. Okay, we'll actually talk about trans fats in just a sec. And the positioning of the orbitals around that bond is what causes the kink. Now, because these kinky little fatty acid tails here don't really stack very easily, these fats tend to stay fluid, they tend to stay liquid at room temperature. So to get a slightly different view of these, what we see here are 3 versions of fats. Here up at top, this is a fully saturated fat. Okay. Right here, this is a monounsaturated fat. In the middle, at the bottom, is a polyunsaturated fat. Up at top, where you see those little oxygens in there, that's actually the glycerol head. It's kind of bent and curled around itself. And then all the zigzag lines are the 3 fatty acid tails. These can freely rotate in space. So it doesn't look like they're stacking on themselves right now, but they definitely do have the capability of doing so. When we look at the monounsaturated fat, notice how there's only one double bond in there, so you only kink once. So you can still kind of sort of stack a little bit, but not as easily as fully saturated fat. A polyunsaturated fat, on the other hand, looks like this down here. It's kind of crazy looking. Has a bunch of double bonds, and because of that, it is highly kinked, and it's very, very fluid. Those molecules move easily past each other rather than stacking. Now, what is trans fat? Remember we said cis versus trans double bonds just a couple seconds ago? So if we recall, maybe from chemistry, a cis bond is where the hydrogens, or whichever functional group you want to point out, are on the same side. A trans double bond is where the hydrogens are on opposite sides of that double bond. And remember, double bonds can't easily rotate around, so they're kind of locked in place. That hydrogen on top of the cis double bond can't rotate around to the other side. So because of that, when you have a cis double bond, it actually changes the direction of the carbon chain. So it's going up, then down, then up again here. With a trans double bond, it doesn't really change the direction all that much. It does a little bit, but not very drastically. So a trans fat actually looks more like a saturated fat, which is kind of a straight line, than an unsaturated fat. And because it looks like it, remember, structure dictates function, it acts more like it too. So trans fats, they have the same mouthfeel as saturated fats, and they tend to be more solid at room temperature. And because of that, as we'll see, they're implicated in heart disease and other diseases like that. Now, how trans fats are actually made, and you don't have to know this is just cool. They're actually produced artificially through a process called hydrogenation. So on the back of a nutrition label, whenever you read the words, partially hydrogenated vegetable oil, it actually indicates the presence of trans fats. And what hydrogenation does is it takes regular unsaturated fats, adds some hydrogens, rearranges some of the bonds, and isomerizing, changing some of those bonds from cis to trans form. And the reason why we do this is because both saturated and trans fats actually have a longer shelf life than unsaturated fats. They're less prone to decay, less prone to oxidation actually. And so obviously they last longer, especially in baked goods, and they feel better. They have a better what's called mouth feel. This is actually part of the reason why fish, which has lots and lots of unsaturated fats, goes bad like within 3 days in your fridge, whereas beef, which has many more saturated fats, tends to last quite a bit longer. Now, why are saturated and trans fats so bad for you? Well, when you eat them and you start breaking them down when you break those fats down into individual fatty acids, they do travel through your bloodstream before they get to your individual cells, and saturated and trans fats can stack on each other very, very easily. They can stack. And because they can stack, they can block an artery actually by building up on themselves. So here we see a plaque in an artery, okay, made of saturated fat and or trans fat. Now this is different. This contributes to high blood pressure, it contributes to blocked arteries, could contribute to stroke, etc. This is different though from the hardening of the arteries. That's where arteries lose their flexible outer coating. Alright, let's go and let's look at our second type of lipid, phospholipids. So just a reminder, we talked about fats, now we're talking about phospholipids. Okay? So phospholipids are important in biology. I mean, all these molecules are important, right? But phospholipids essentially, they are the big component, the major component of all cell membranes. And there's actually many different types of phospholipids, not just one. And different cells will have different types of these phospholipids. When we look at a phospholipid, it looks a heck of a lot like a fat. Remember, a fat had 3 fatty acid tails and a glycerol head. Well, a phospholipid will have 2 fatty acid tails, a glycerol head, and an additional phosphate group. If we remember, a phosphate group is PO4. Okay, so phosphate group is PO4. And when we look at the structure of a phospholipid, it looks something like this. Alright, so here's a phospholipid, here's the fatty acid tails, right here and right here. One of those fatty acid tails is saturated, one is unsaturated. It's actually polyunsaturated because there's 2 double bonds in there. Here's the glycerol head up here, carbon number 1, carbon number 2, carbon number 3. Here's the phosphate group. Alright, and there's some other stuff that you can add on there as well, and this is where the phospholipids will differ from each other in what other stuff is added on. And we can also represent phospholipids not only chemically with this structure, but also as this little weird figure over here with the head and the 2 tails. Now the thing about phospholipids is those 2 fatty acid tails are actually hydrophobic. They're made mostly of carbon and hydrogen. Alright? So those 2 fatty acid tails are hydrophobic, but the phosphate right here and this other group are very, very hydrophilic. Alright? And that other group, in this example, it happens to be a molecule called choline. So this molecule right here kind of has the best of both worlds. He's both hydrophilic and hydrophobic, a little bit of both. If we look at a fat molecule, on the other hand, go back to our fat, it is almost entirely hydrophobic. So that's what makes phospholipids special when they interact with water, that half hydrophilic, half hydrophobic nature. So here we see two layers of phospholipids, and I'm going to draw out the head as the ball, so that's the glycerol head and the phosphate group. And then the fatty acid tails as the little squigglies down here, two fatty acid tails, therefore two squigglies. When you put a droplet of phospholipids into water, their heads will face inwards and outwards towards water, whereas their tails will kind of group together, trying to get away from that water. Those hydrophobic tails, they don't like water. All right. So most of the time, these phospholipids will form what's called a bilayer. So a bilayer, bi means two layers, so that the heads can point outwards and the tails can point inwards. And those phospholipids, they'll actually form a circle with water inside and water outside as well. So when we have something like this, a little fatty membrane, okay, with water inside and outside, we're starting to look an awful lot like an early cell. And in fact, phospholipids, as we just said, are actually the majority of our cell membranes. Let's look at our third major type of lipid, steroids. Steroids. Alright. So steroids are not just, you normally think of like anabolic steroids like roid rage and all that. Steroids are actually a class of molecules that do include anabolic steroids. So chemically speaking, steroids are lipids. They're not made of fatty acid tails though. Okay, they don't have glycerol heads or anything like that. But they are made of carbon, almost exclusively carbon and hydrogen. So here's what a pretty average steroid looks like down here. Okay, this is cholesterol down here. And as we can see, cholesterol is made of 4 rings. And at each apex here, there's a carbon, 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 all right, along with hydrogens. So and the rings share a size, so they are fused together. And because this molecule is mostly carbon and hydrogen, it is also very hydrophobic. And that's what a lipid is, it's just a hydrophobic molecule. Cholesterol is an important component in our cell membranes, actually the cell membranes of all animals. And what cholesterol does there is it acts as what's called a fluidity buffer. A fluidity buffer. So it helps the cell membrane be a little looser, and at the same time it helps it be a little more rigid. Because if your cell membrane were made just of phospholipids, it'd be about as fluid as solid oil. And obviously we are not walking puddles of oil, right? So we need some stiffness to our cell membranes, and cholesterol helps to stiffen up our cell membranes. Now if you're a plant though, you don't have cholesterol, you don't really need it. You have instead a nice thick supportive cell wall made of cellulose. So plants are actually do not have cholesterol, and therefore when you eat plants, you do not get cholesterol into your diet, which is not necessarily a bad thing, right? Now, besides cholesterol, other steroids, one of their major functions is to act in cell communication and trigger those cells to grow and to develop into different cell types. And part of the reason why is these steroid hormones, as they're called, these other communication molecules, these steroid hormones, can very easily get through cell membranes. So they can easily slip through those phospholipid bilayers into the cell and trigger the cell to change its behavior. So let's take a look at some of those steroid hormones. Here's two examples, estradiol and testosterone. So estradiol is actually a form of estrogen. So estrogen and testosterone are both steroid hormones. And as it turns out, they're actually derived from cholesterol. So our cells can make cholesterol, some of them anyways, and we can convert that cholesterol using a certain class of enzymes into both estrogen and testosterone. And estrogen and testosterone, they don't do the same thing as cholesterol at all. They have a different structure. Notice these OH groups the carbonyl, the C double bond O, the CH3 that we've added over here, because they have a slightly different structure, they will do totally different things. So whereas cholesterol stiffens cell membranes, estradiol or estrogen and testosterone will trigger cells to divide, it'll trigger cells to specialize and to change what genes they express and what genes they don't. We're actually going to come back to these when we talk about cell signaling and talk about how steroid hormones work. Now I know some of you guys, we got to talk about the elephant in the room, right? Steroids in medicine. So when we talk about steroid use in medicine, one of the major types of steroids that we use are called corticosteroids. Corticosteroids are actually a type of signaling steroid, so they're kind of similar to estrogen and testosterone. They act to assist in cell communication. And one of the things that corticosteroids do is they actually suppress the immune system. So corticosteroids suppress the immune system. And because of that, they can be used to treat inflammation. They can be used to treat immune diseases. So they're used to treat like rheumatoid arthritis. They're used to treat lupus, which is an immune disease. They're used to treat like really, really bad sunburns where your immune system may go haywire because you've caused so much damage to your skin, etc. So they are used to treat a huge array of diseases and symptoms. Now, their side effect is, you get sick a lot more easily because they do suppress your immune system. Anabolic steroids, on the other hand, are another class of steroid. They look like testosterone to a cell. So when you inject anabolic steroids, a cell thinks it's just getting a burst of testosterone. And what testosterone tells cells to do, remember we said it was a signaling molecule, it tells cells to up their rate of protein synthesis. So cells make a lot more proteins, especially if they are muscle cells. So muscle cells are really, really sensitive to testosterone. Now what that means is your muscles get bigger and packed more full of protein, right? It'll also act on cells that produce hair, and it'll act on other cells, so you not only get increased muscle mass, you also get some secondary male sex characteristics, like increased hair growth, etc. But what happens is that excess testosterone, because remember your cells think they're getting lots and lots and lots of testosterone, is actually converted into estrogen. So your cells are going to go, sweet, I got testosterone, let me convert some into estrogen because as a guy you actually need some estrogen too. Now if you go off those steroids though, suddenly your own natural production of testosterone can't keep up, and you've got a relatively low amount of testosterone and a lot of leftover estrogen, which means that you get female secondary sex characteristics, and one of those female characteristics is gynecomastia. And I actually misspelled this. Gynecomastia, also known as man boobs, right? So, besides gynecomastia, you can also get some other nasty side effects like testicular atrophy, reduced sperm production, etc. So, don't do steroids, kids. Alright, I'm going to hit pause here and I'm going to actually upload this as a separate video and start nucleic acids as its own.